This is Holy Cross Mock Component 2 and it's paper 4 and we are on to question 5. Hopefully this is a bit of a break after the Warfarin question. Nice, straightforward 7 marks hopefully. Don't worry, it's not your eyes, the print is very faint, but I'll read most of it out. Some human DNA was cut into separate pieces using a restriction enzyme which produced a staggered cut. So here's, let's do it down here. Here's some DNA and it's cut using a staggered cut. There's a staggered cut and here's another staggered cut. And this is our gene there. So when we take that gene out, it looks a bit like this. So I'm just going to draw the bit in the middle. That's what our gene looks like. So that's come from there. That's that piece there. And it says insert these pieces into plasmids. So a plasmid is a circular loop of DNA. I'm not going to draw it all, I'm just going to draw this bit of the plasmid and I'm going to do the same staggered cut. There we are, same staggered cut. And so that would leave this sort of shape. So this bit here, there we are, and then on the end bit, or the other bit there, would be sort of that bit, and then that'd go there. So that's where we've cut, and then we're just going to pull it apart, and then that's great because that bit can then go in there. And that'll just fit absolutely bob on there. So that's our stick to the same colour. That's our gene there. So it'll fit in, and it fits in there. And these are complementary bases. And that's what we call a sticky end. And explain why the pieces of human DNA will be able to join. So we are just going to say sticky ends are formed. Or you could describe that picture and this leaves, exposed bases, which will bond to complementary bases. Or a description of what you've got there. Which other enzyme must the scientist have added to the mixture to form recombinant plasmids? So these sticky ends will fit in and the complementary bases will bond by hydrogen bonds. But that's not enough. There's the bonds actually between the nucleotides, so we need bonds there. And those are made by an enzyme, a ligating or joining enzyme called ligase. So DNA ligase. And ligase is the important word there. A plasmid may be used as a vector. So here's the plasmid. So if I draw a little version of it there, and you can pop that into a bacterium. You can persuade it. You can treat bacteria in various ways, various chemicals that allow plasmids to enter, and then the bacterium will then hopefully transcribe and translate that gene and produce the relevant protein, whatever useful protein it is. So the vector is used to transfer that plasmid, and it's got our useful gene in there, into another organism. So it's the carrier of the gene. So it carries the DNA or gene of interest into, and then the description of whatever we're going to put it into, into another organism, for example a bacterium. Molecular biologists often use plasmids that contain antibiotic resistance genes. So in addition to this gene, then they will stick in another gene for resistance. And it's just a way of seeing whether this plasmid has been taken up. Because if the plasmid has been taken up and it's resistant to an antibiotic, if you treat our bacteria with this antibiotic, they will survive. If they die, it's because they haven't got the resistance gene, but then they also haven't got our gene of interest, like an insulin gene. So it's just a way of seeing whether the plasmid's been taken up. So the technical word is a marker gene for this. So it's a marker gene. And it allows us to detect the cells that are actually containing our DNA or a gene that we've added. So allows detection of cells containing the plasmid with the gene of interest, or the DNA, or the recombinant DNA. Various ways of saying that. And if we've got our kind of 2 marks there, but we explain, well, and resistant to what. So will survive, or you can do the other way, or die. Will survive if bacterium given the antibiotic. So just as you mentioned, the antibiotic and anything with the resistance gene will survive. And that's hopefully a fairly straightforward 7 marks.